Okay, let's get started. It's my pleasure to introduce Dr. Emily Maraldi, who's a joint faculty candidate between infectious disease and DCMB. And I think her joint recruitment by these two departments is really a reflection of her being a true interdisciplinary scientist. So she got her PhD from MIT in the computational biology and systems biology program in the lab of Forrest White, where she applied experimental and modeling approaches to decipher in vivo phosphorylation networks. From there, she went on to do her postdoc at NYU, and she's joined between the labs of Richard Bonneau and Dan Littman, who are respectively aficionados in systems biology and immunology. And I think bridging those two worlds has a lot of exciting potential. And Emily's been doing work, working with computational experimental scientists during her postdoc to generate really exciting data sets and apply novel analytical techniques to try to reconstruct ecological and molecular networks underlying host microbe interactions. So with that, I'll let Emily do the rest. And thank you so much for coming. And we're very excited to see your talk.
Thanks, everyone, for being here to listen to my talk. I'm really excited to be here. And so just to start off, I'd like to introduce the gut ecosystem. So here we have a cartoon. And just to orient you, the top we're looking at the lumen, so the contents of the gut. And then on the bottom, we're looking at the lamina propria, the lining underneath the gut. And then these are a line of epithelial cells that separates you from your microbial lumen content. And one of the reasons I like this figure is because you don't have to worry about the names on it, but there are a bunch of different immune cell types represented. And there are numerous microbial taxa that are represented. And then something that makes this figure especially nice is that they actually represented some heterogeneity in the epithelial cells too. So this ecosystem has a lot of different components. And it turns out that alterations in this ecosystem can contribute to the development of autoimmune diseases. And so as a specific example, I'd like to introduce the microbe segmented filamentous bacterium, which I'll abbreviate as SFB. And it was discovered as one of the first examples of a commensal gut resident microbe that played a role in autoimmune disease. And so here we're looking at a false colored image. And you can see the small intestine villi sticking out here. And the SFB is a segmented, long filamentous structure that's in very close contact to the epithelial cells. And it turns out that these mice have interesting phenotypes. And these microbes can induce changes in immune cell populations. So here, for reference, is another picture of the gut. And we have, this is for SFB negative cells, so mice lacking in SFB. We have a couple of cell types. These are dendritic cells. They have long dendrites that can stick into the lumen and actually sample the contents. There are Th17 cells, ILC3s, which you'll notice there are just a couple of Th17 cells in this figure. And when you have SFB as a member of the gut microbiome, it leads to an expansion of these Th17 cells. And these Th17 cells are associated with pro-inflammatory responses, and they are important for fighting pathogens, in particular microbes and fungi, but they're also implicated in autoimmune diseases, so they contribute to different autoimmune diseases. And one of the things that I'd like to just to summarize here is that if you start off with mice that don't have SFB, and then you give them, say, a fecal gavage of containing SFB, the process of induction of Th17 cells involves multiple host cell types, takes place in multiple body site locations, different parts of the intestine, involve mesenteric lymph nodes, and then the expansion of Th17 cells unfolds over time course. And so at early time points, you don't have these Th17 cells, but unique responses in some cell types. Later on, you get an expansion. And so this is a complex process involving a number of different cell types. And it leads to some interesting phenotypes. So SFB positive mice, they are more likely to fight off pathogenic infections. For example, Citrobacter infection, which is a mouse model of E. coli food poisoning. But they're also more likely to develop mouse models of autoimmune diseases, including inflammatory bowel disease, and also multiple sclerosis and rheumatoid arthritis involving the central nervous system or the joints. So autoimmune diseases that are not proximal to the gut, but actually further away. So this particular microbe, through its induction of Th17 cells, influences host systemic immunity. As just one more quick example of another gut microbe that has an immunomodulating effect, I just want to introduce Helicobacter hepaticus. In wild-type mice, presence of Helicobacter promotes an anti-inflammatory response. But if you look at mice with inflammatory bowel disease-associated mutations, for example, ablation of IL-10 signaling, these mice actually develop spontaneous colitis, which is a model for inflammatory bowel disease. So these are two examples of microbes that affect host immunity. And interestingly, for both of these models, there have been studies that have shown that not only does it matter that the presence of the single microbe is in the gut, but it matters what other microbes are present too, suggesting that the whole ecosystem contributes to the mouse phenotypes. And so when we're thinking about a cartoon of SFB, this is a little bit ugly and scary, but you should be thinking about the whole ecosystem and also many different types of immune cells. And so when thinking about trying to study the system, it's almost overwhelmingly complex. And when you have a system that involves a lot of different interacting parties, one great source of scientific tools comes from engineering. And mathematical modeling frameworks have the sophistication of being able to describe this kind of network. And so I'd like to describe to you an R01 grant that I wrote with my two advisors to study the gut ecosystem. And so the design of the study involves a perturbation time course experiment, where at time 0, you stimulate induce a cytokine blockade, cutting off either IL-23 or IL-10 signaling. Both of these signaling pathways have previously been associated with human inflammatory bowel disease via genome-wide association studies. And so you kind of layer onto these mice a genetic defect that has been known to influence autoimmunity in humans. And then 2 weeks later, you layer on a microbial perturbation, adding SFB, H. hepaticus, or control. But these two microbes, of course, have been associated with autoimmune disease already. So this is kind of an experimental model for how an autoimmune disease might develop due to genetic predisposition for an autoimmune disease layered on, with this case, a microbial perturbation to cause autoimmune phenotypes. And so this is the time course. What kind of measurements are we hoping to make, and what kind of models do we want to get out of it? And so as far as measurements, we're interested in measuring immune cell populations using flow cytometry. So to figure out which populations of immune cells are changing, we're interested in 16S rRNA DNA sequencing to get microbial compositions to see how the microbes are changing over the course of this experiment. And then we're also interested in measuring host responses at the molecular level using RNA-seq and ATAC-seq technologies, which I'll describe in more detail later. We're interested in multiple host cell types, looking at different body sites. And then for me, the way that I'm really excited about putting this together is in a mathematical model. And so we're interested in 2 levels of mathematical modeling. One, we want to be able to predict the evolution of microbial and immune cell populations. First of all, we'd like to be able to describe it over the course of this time course. and then be able to do computational experiments where we can say what happens if we, say, remove some of these microbes or immune cell populations, how would that change the evolution of the system? And the second aim is to build transcriptional regulatory models of immune cells responding to these perturbations in vivo. And so I'll go into more detail for both of those. But one of the things I wanted to focus on, although these goals seem really different, population level models, molecular level models, and they do have different challenges, they do share some similarities. So data from both of these models are in what's known as the underdetermined regime. So I have two data sets. This is a data set of microbial abundances. We have 210 taxa that are measured, 100 samples. Here's another data set, and we have gene expression measurements, so about 5,000 genes and 100 samples. And what you can see in both of these cases is that you have more measurements than you have number of samples. And if we're trying to, say, infer ecological interactions, then we actually have many, many more interactions or parameters to be estimated the number of samples. And so this leads to some practical implications. Number one, seem to be too many parameters to estimate. And so sometimes it's not possible to find a solution, or sometimes it's possible to find many solutions, and a lot of those are going to be wrong. And so, you know, if you're working in this regime, you're at a very serious risk of coming up with spurious results. And so just to give you a basic framework and appreciation for some of the inference methods that I'm using, I wanted to point out the general strategies that I'm using. So you start with an underpowered data set, be it microbial abundances, gene expression, and you want to get out a set of accurate models. And so one of the things that we do in our inference procedures is we try to integrate external data, prior knowledge, and then another thing we try to integrate are assumptions. And just as an example, one assumption that we make is that in both microbial ecological networks and transcriptional regulatory networks is that the underlying set of interactions are sparse. And so here I've illustrated this with a picture of two toy microbial networks. And the basic idea, in contrast, here's a dense network where all of the microbes or populations are interacting with each other, represented by lines. And then sparse network. And so this is our assumption when we're building these networks, that on average, for example, if we're thinking in terms of microbial networks, your average microbial taxa doesn't interact with most of the other microbial taxa, but maybe just a handful. And so just to move on and show you a little bit about what I've done in the domain of ecological interaction network inference. One of our first challenges is to build, and the goal here is dynamic population models of microbes and immune cells in the gut. But one of the problems is that a lot of these microbes, for example, are known only on the basis of their sequences, and they can't be cultured externally. And we don't actually know what the ecological interactions are. So if there's a mutualistic interaction, if two microbes are in competition with each other. And so one of the first challenges to even building a dynamic model is actually figuring out what are the ecological interactions. And so just to phrase the goal more clearly, we have a table of OTU, stands for Operational Taxonomic Unit. So that's a level of microbial taxonomy. So you can think of this as a microbial taxa versus samples. And we want to come up with a computational method that tells us what are the ecological interactions among those OTUs. And here this is represented as a graph where the nodes or circles represent microbial taxa, and then the edges represent an interaction. So that's the goal. And I did this work in collaboration with two computational biologists in the Bonneau lab, Zach Kurtz and Christian Mueller. And I'm going to just very, very briefly go through what I think are the key points. But this work is published, and I'd like to focus more on the transcriptional regulatory networks because that's not published. So I'm just going to summarize briefly here. Our method is called Speakeasy, and it stands for Sparse Inverse Covariance Estimation for Ecological Association Inference. And again, the inputs and outputs are known. There are two parts to the software that we've released. The first is a network inference pipeline, where we learn a sparse Gaussian graphical model. And I'm happy to talk about more of these details afterwards or in my chalk talk tomorrow. The other part that I'm very proud of is a realistic synthetic data generator. And one of the challenges with inferring ecological interactions is there are no experimentally validated large-scale ecological networks. So you can come up with a pipeline that you think is going to be very good, but how do you actually know it's good? And so one of the major challenges we had with convincing ourselves this method was good was being able to generate realistic synthetic data, specifically correlated over-dispersed counts data from models of known topologies. And so one of the things that you can do with this method is you can actually generate synthetic data that matches, in a statistical sense, your input data set and be able to see how well this method recovers on your particular data set. Okay, and then this particular, so the output is an interaction network, and as is done by Pat Schloss and Pat Schloss's lab in previous publication, it can be used as kind of a skeleton or a scaffold for building a more complex model, because it can tell you which parameters to focus on if you know a priori that some interactions are present and some are not. So you can reduce some of the parameters that you have to estimate. So this would form the backbone of dynamic models. And currently in the Littman lab, we're working on collecting the data set for the ecological models. And so now I'd like to describe in pretty gross detail the transcriptional regulatory networks that I've been building. And so the Bonneau and Littman labs have a history of collaborating together to build transcriptional regulatory networks in immune cell types. And the first collaboration was to develop a network of Th17 cells, which I already said were important cells in protecting the body against pathogens, but also implicated in autoimmune disease. And this was a very serious collaboration in that it involved the generation of a huge data set. So there were 11 TF-Chip-seq experiments. There were 28 transcription factor, TF is abbreviation for transcription factor, knockout experiments, about 150 RNA-seq experiments. And then in order to make this network, they also included this microarray immune cell compendium gene expression data set called ImGen, and came up with this final network. And this scale of a project was really possible because Th17 cells had been studied to the extent that we knew the conditions for developing Th17 cells in vitro in tissue culture. And so, although inspiring and a great proof of concept in a lot of regards, this is very different from what's proposed in this grant. We're interested here in a physiological setting and looking at responses to these perturbations. So this is in vivo transcriptional regulatory networks. And so off the bat, we know that we cannot generate the scale of data sets because sample is going to be limiting in this context. And so let me show you a little bit more. But one of the first things that we decided to focus on was to infer transcriptional regulatory networks in a newly discovered, last 5 or 6 years, immune cell type known as innate lymphoid cells. And this is in collaboration with Maria Prokofsky and Jason Hall in the Littman lab. And I'm not going to go too much into the immunology, but these 3 innate lymphoid cells, ILC subtypes, they live at mucosal interfaces, and each of them play unique roles in homeostasis and also have been implicated in different autoimmune diseases. And just like to highlight ILC3s because they're actually involved in the induction, an SFB induction of Th17 cells, which I mentioned briefly. importantly, as an exciting research topic, little was known about their gene expression regulation, so there's a lot to learn. And one of the challenges is that these cells are relatively rare. So there are only about 10 to the 5th of these cells in the small intestine of a mouse, for example. And it's not known how to expand all 3 of these cell types in tissue culture, so they really do have to be studied ex vivo. Before I describe the mathematics of transcriptional regulatory network inference, I just want to show you in cartoons the kind of model that I'm trying to build. And so here we have a model of cis gene regulation. Here's a gene locus, and we have bound cis to that gene, or bound cis to the gene locus, different transcription factors. They're nearby. They might be playing a role in regulating this gene's expression. And in another cell type, you might expect to have some of the same transcription factors involved, but maybe some different transcription factors as well. And key to genome-wide figuring out where transcription factors are bound across the genome have been ChIP-seq experiments. And so here's a cartoon of the kind of data that you might get from a ChIP-seq experiment for this blue transcription factor. And here you see a problem. Each of these experiments requires about 10 to the 7th cells, about 2 orders of magnitude more than are present in a mouse gut. And really, if you wanted to build a high-quality genome-wide transcriptional regulatory network, it would be nice to have data for all the relevant transcription factors. So really not feasible for innate lymphoid cells. And so the breakthrough in technology that really enabled this project is a new data type known as ATAC-seq, assay for transposase accessible chromatin. And I'm just going to show you in cartoon form the kind of data that you get from this assay. So it basically shows you what part of the chromatin is nucleosome-free or accessible. And you can see that these peaks correspond to where transcription factors are bound. So there's a good correlation between these nucleosome-free regions and transcription factor binding. But the problem here is that we don't know which transcription factor was binding in that accessible region. And so the missing piece of information is partially filled in using motif data. So different transcription factors have different affinities for particular DNA sequences. And so by scanning the DNA for motif occurrences, you can implicate a TF with particular peaks. And so this is how you can get ChIP-seq-like information out of a single experiment through integration with these motif databases. And so the idea here was then to combine gene expression and ATAC-seq data to infer transcriptional networks in ILCs. And the mathematical framework that we're using is sparse linear regression. So basically, for each gene, we model its expression as a multivariate combination of the transcription factor activities that regulate it. And these coefficient betas can be positive or negative, depending on whether that interaction is activating or inhibitory. And we can solve this particular problem given a data matrix X, transcription factors A, and solve for this matrix of coefficients using a number of different methods. The Bonneau Labs inference method is known as the Inferelator, and our current method uses what's known as Bayesian best subset regression. And so this is published, it came out, I think, in 2013. And so it uses a couple of tricks to get to a limited set of potential regulators of a particular gene. And so one of them is this method enables you to incorporate external information. So if you happen to have information from a database or some other source and you know that a transcription factor does indeed regulate a gene, you can incorporate that by including it in, we use a G prior, which will then bias the model to be more likely to explain a gene's expression pattern with the transcription factor that's known to influence it already. And then we are able to make sure that we are able to incorporate the sparsity assumption using Bayesian information criteria for model selection. And basically, the Bayesian information score is a balance between model fit and complexity or number of parameters in the model. So by calculating this Bayesian information criteria, we are able to get a sparse fit of the data. And so this kind of gives you an idea of how we go about building, except for I left out one of the most important parts. The predictors. What actually are transcription factor activities? And last year, people were estimating transcription factor activities looking at the transcription factor's mRNA levels. And of course, this is imperfect because a lot happens between being a piece of mRNA and being an active transcription factor. And if we really wanted to make good predictions, it would be great to have some, say, protein-based assay of transcription factor activity. But usually we don't have that data. And in that case, transcription factor activities are really, you think of them as hidden variables. So you know the transcription factor's gene expression level, something happens, and the active transcription factors, are unmeasured, but are influencing gene expression. And so other groups have suggested that you can estimate a transcription factor's activity by looking at the expression of its target genes. And so here we have another equation where you have a matrix of the target genes, and then you incorporate information from prior, and basically, so the prior is basically a table where you have transcription factors versus target genes, and a 1 would indicate that you have a positive interaction, 0, no interaction, -1, an inhibitory interaction. And then they're actually, this doesn't have an exact solution, but using least squares approximation, you can solve for A, the activities, and then plug back up in here, and then use the Inferelator BBSR to solve this. And this idea ended up working great in B. subtilis. And so B. subtilis is a really well-studied model organism, and so there's a database with thousands of known transcription factor gene interactions. And this has been recently published in MSB. And I will say one of the cool things from this paper, this prior, it's based on a database of high-quality interactions, but they also did experiments in this paper where they mixed into the quality prior matrix false interactions and tested on a leave-out set. And they found that actually the method was pretty robust, even if you outnumbered good interactions to random false interactions 10-to-1. And so it's always an improvement, regardless of if your prior is perfect, in our hands to actually calculate transcription factor activities instead of just using the mRNA level. And so this seems very exciting, but I don't have a database like this for innate lymphoid cells. There are about 20 interactions that are known. And so this is where the ATAC-seq data comes in. And so the basic idea was to use this epigenetic data to get that prior matrix of transcription factor gene interactions. And just to be a little bit more specific, when I was building the prior, and this is what I call the simple prior, I would associate a transcription factor with a gene if that transcription factor's motif fell in an accessible region that was plus or minus 10 kb of that gene. So a little bit arbitrary, but that's how I built the prior matrix. And then the idea, of course, is then to take that prior, combine it with a gene expression data set, run now what I call Inferelator BBSR TFA, and come up with an ATAC-Inferelator network. And just to give you a little bit of a sense of how using a prior and transcription factor activity estimate looks like, here we go back to the Th17 data set. And we have at the top this Western blot that shows protein levels across 0 to 96 hours. And below, we have matched microarray gene expression data from 0 to 96 hours, so the same time course. And below, we have transcription factor activities as estimated from probably our best type of prior based on chip and knockout data. And in particular, I just wanted to point out that STAT3 activity correlates very nicely with phosphostat3 abundance. And phosphorylated STAT3 is a proxy for STAT3 activation. And it looks a lot better than, say, looking at just the mRNA level. And so returning back to here, we've made an improvement in prediction by basically improving our estimation of our predictors. And so this seems like a great pipeline, at least it did to me. But there was a serious concern that both me and my collaborator had, and she was as serious about, this seems like a good pipeline, but how do we know if it's good? And so what she did was she basically also generated 33 ATAC-seq experiments to benchmark. And these were done in Th17 cells for which we had all of this data already. And so we could start to benchmark this method. And so I'd like to show you some of the benchmark results before I go on to show you the results for the innate lymphoid cells. So the first question is ATAC-seq data, a single experiment, as useful as 11 ChIP-seq experiments in key Th17 transcription factors? And so here's the pipeline that we came up with to make this comparison. We could, analogously to the way we developed an ATAC-seq prior, we could come up with a ChIP prior based on those 11 ChIP-seq experiments. And then as gene expression input, we actually had 3 different gene expression data sets that we used. And for each of those data sets, we could get a ChIP or an ATAC-seq network. And then in order to basically decide whether or not we were performing well, we could look to see how well it corresponded to a network constructed from the knockout data. So if you knock out a transcription factor and you see a change in a gene, that's at least supportive that that transcription factor might be influencing that gene's expression. This gold standard is not perfect because, of course, knockouts include indirect interactions, and you also have network compensation. So you might not see a change in a gene, even if it is affected by a particular transcription factor. So there's some caveats. But all in all, we would expect, we'd feel good if these networks had some correspondence with the knockout data set. And then the final idea was once we were satisfied with the ATAC versus ChIP-seq comparison, then we could go ahead and compare the ATAC-seq networks to probably a better gold standard, a gold standard consisting of transcription factor gene interactions that were supported not only by knockout, but also by ChIP data, suggesting that that transcription factor also bounds cis to those genes, so more likely to be a regulator. And so we evaluated performance in a number of different ways, but the one that I'll show you here is precision recall. And so basically a predicted network is just a ranked list of transcription factor gene interactions. And so what you can do is you can move down this ranked list of interactions and calculate your precision, the percent of predicted edges that are actually in your gold standard. And you can also calculate the recall, the percentage of your gold standard network that you've actually captured at that particular cutoff. And so here are some examples of precision recall curves. And you can imagine having a list of high confidence interactions to low confidence. And as you move down the list, your precision drops off and your recall would increase. And here, just for comparison, you can see that this method, whatever it is not performing as well. Its precision drops off very quickly. And so what do precision recall curves look like comparing the ChIP and ATAC-seq networks? And so here we have precision recall. This is the ATAC ChIP. And this is comparison to not using a prior at all, so the old Inferelator method, and this is random background. And so roughly speaking, and I'm only showing you results for one of the gene expression data sets, we saw that CHIP and ATAC performed more or less similarly. So they seem to both do, both represent an improvement over not using a prior. And one of the second questions we were interested in asking when we were doing this Th17 benchmarking was, well, what if you didn't have any ATAC-seq measurements that were done, for the same experimental conditions? Could you pull in more general resources? And so we constructed two what I would call generic priors. And the first was based on this trust database, which was generated using machine learning algorithms to basically text mine and find articles that seemed to indicate a transcription factor gene interaction. And then they were manually curated by a group of 30 people. And so it's actually a pretty high quality database. I was surprised by the quality of interactions that were in there. And you'll see that it actually performs pretty well. The other generic prior that we used was based on DNase hypersensitivity accessibility measurements in 25 different mouse tissues. That was part of the ENCODE project. And basically, footprinting was done in these accessible regions. So this is kind of similar to what I'd done, except for in random cell types that were not Th17. And what we see is that these methods help too. And if you then go ahead and combine these different priors, you can get even better performance. So some of the top performing priors were a combination of ATAC-seq and trust and ATAC-encode and trust. The one last Th17 benchmarking comparison that I want to show you is, so I told you about how I generated a simple ATAC-seq prior, but up until now, I haven't shown you any ATAC-seq data. So one of the cool things about these data sets is they are quantitative, so you can treat them in a quantitative manner. Here, this is a heat map, and we're looking at accessibility of 170,000 peaks that were detected across these T helper cell samples. And here's Th17 time course, and we have other T helpers that were controls in this experiment. But you can see that they have very different trends. And so there's some quantitative information there. And here I've just picked a random transcription factor that's expressed in Th17 cells, RXA alpha. And all of these clusters are enriched for the RXA or RXR alpha motif. And what I found striking was that so some of these clusters show decrease in accessibility over the course of these differentiation time course, suggesting that RXR alpha or whatever is contributing to that peak is binding maybe less strongly over the time course. But then we also see enrichment in clusters where the accessibility is increasing over the course of the Th17 time course. So the explanation here, so we have different, we have motif enrichments for the same, or enrichments for the same motif, but in clusters with different patterns. So they're probably due to different biological phenomena. For example, maybe the orange one is due to binding of RXR alpha. Maybe the green one's due to RXR beta, an RXR family member that has a degenerate motif. Or maybe they're due to differences in the proteins that are heterodimerizing with RXR alpha. And so whatever the source of this reason, this gave me the idea to try to take advantage of the quantitative nature of the ATAC-seq data. And so I generated what I call a quantitative ATAC-seq prior. And basically, I'm going to depict it here, use it just in pictures. And so for each expressed transcription factor, instead of just looking to see whether there was a peak that was cis to a gene, I looked at gene clusters, so say genes that were just increasing under Th17 conditions. And then I checked to see whether peaks that had particular patterns, say peaks that were increasing in accessibility under Th17 conditions were enriched cis to these genes. And if they were, then I added an edge in the prior connecting that TF to the genes. And so this is another way of generating a prior. And here I'm going to show you results comparing to the better gold standard, the knockout chip. And you can see that when you include the quantitative information in combination with some of the generic priors, you do get a little boost in predictive power. And precision recall is one good way to look at these networks, but it's also very nice to look at them in graphical form. And so in collaboration with a very talented software engineer at the Simons Foundation, Aaron Waters, I helped to design an IPython notebook platform for network visualization, where we were able to compare, say, this is the knockout chip gold standard. These big hubs correspond to the 11 transcription factors that were chipped. And you could compare it to the inferred ATAC-seq network. And you could sync the network so you could compare between the two. I'm not going to show you too many features, but one of the things that we did to gain confidence in our method was to compare the key Th17 genes and transcription factor regulators to between both of the networks. And what we see in general is that the ATAC-seq networks tend to be sparser, so have fewer predictions than knockout chip. But we're hoping that these networks become denser as we integrate more data sets. So we're still in the building phase of combining more data sets. The other last thing I want to tell you about the Th17 network, just to emphasize, these networks are made only of transcription factors. And you can see here's chip where you're limited to the TFs that you chip for. And here's ATAC-seq. And so you can see that your coverage of the transcription factor network greatly increases. And in stars are the transcription factors that my collaborator, Maria Pokrovsky, thought should be in the Th17 network. The other ones are candidates that have not previously been associated with Th17. And so just to summarize here, the things that we learned from this Th17 benchmark are that chip and ATAC-seq derived priors do improve performance of network inference in the mammalian setting. Generic priors help. And then finally, it does seem reasonable to combine ATAC-seq and gene expression measurements in new cell types that haven't been previously explored. And so with this benchmarking in hand, we went on to build networks in innate lymphoid cells. And here are some of the networks that we recovered. So for shared and unique ILC networks, so ILC1s, ILC2s, ILC3s. And then these are transcriptional regulatory interactions between TFs and genes that are shared between ILC1s and ILC3s, shared between ILC2s and ILC3s. And what we were pleased to see was that for the few master transcription factors that were known in these innate lymphoid cells, we were able to recover them. And what was also very exciting is that we recovered 43 new transcription factors. And across these networks, there are 6,000 interactions. Here, just to zoom in, previously for ILC3s, ROR gamma and TCF7 were known to be master transcription factors. You can see that we've expanded the number of regulators, and these are the key, some key ILC3 genes that we were pleased to see were in our network. What are we doing with these networks? So we have a plan for experimental validation. And one of the cool things about generating networks, as opposed to just figuring out which transcription factors are key, if you have a network, you can actually figure out what physiological role the genes that a transcription factor is regulating are actually involved in. So you might know what kind of phenotype to look for if you have a transcription factor knockout. And here, unfortunately, the experimental validation is going to be a lengthy process. because we're not able to culture these cells in vitro. And so basically we have plans for several candidate transcription factors. We're going to use CRISPR interference to knock down transcription factors in ILC progenitors and then bone marrow transplant them into mice and see how the different ILC subtypes develop from those TF knockdown progenitors. And just to kind of bring us full circle, so these networks that I built were all at steady state. But of course, at the beginning, I said that I wanted to build networks of immune cells responding to microbial perturbations. And so recent data that we have, simply an N of 1, so don't pay too much attention to the genes that are listed here. But we have basically done some experiments in ILC3s. We have some time course experiments. So we have 3 days post fecal gavage with either SFB positive fecal material or SFB negative. And then we also have some steady state measurements in SFB monocolonized mice and germ-free. And we, on the basis of other experiments, decided to separate the ILC3s on the basis of surface markers. And we see that the SCA positive and the SCA negative CCR6 positive ILC3s have specific gene expression patterns in response to these different perturbations. And so one of the things, so we don't have ATAC-seq data for this data set, but we do have the ex vivo steady state ATAC-seq data sets. And so what I've done is I've taken the ILC3 networks that we have and basically expanded them so that they could explain the gene expression patterns that we're seeing in the figure before. And these networks, I would not also, again, not focus on the genes too much because it's an N of 1, but this is the basic idea and progress towards the big goal. Although I've showed you ILC3s, I'm also working on a couple of other cell types. I'm doing similar experiments with ex vivo ATAC-seq and RNA-seq measurements. Let's see. As far as progress on the ecological modeling, we're still optimizing time points and exact conditions, but this is part of the grant that I'm hoping to take with me and work on here when I start out. And then just to summarize, these are the strategies that I've used here to build more accurate transcriptional regulatory models. But there are a lot of different ways that you can improve these transcriptional regulatory networks. So these are some of the things that I would like to do to further constrain the model building process and come up with more accurate models. So first of all, if you know something about the architecture of the network, for example, if it's scale-free, or how many transcription factor targets your average transcription factor could have, anything about the network topology, there are methods that are being developed that enable you, if you know something already about what kind of network architecture to expect, you can actually model that and use that to constrain. Another thing that I'm working on with others in the Bonneau lab is to learn networks simultaneously. If you, for example, the ILCs, each of the subtypes has unique and shared regulation. So instead of learning each of those networks separately, to try to share information between all three using what's known as a fusion penalty. And I can talk more about that afterward or at my chalk talk tomorrow. Another way to improve these, so I've actually thrown out a lot of the interesting or differential ATAC-seq peaks because they weren't plus or minus 10 kb of gene. And of course, and so incorporating 3D chromatin information would be another way that we could improve these transcription factor models. And then other ideas, we're limited here to transcription factors, protein transcription factors, but it would be very cool to consider non-coding RNAs, and also maybe to explicitly model complex interactions. So there are a lot of different ways that these networks could be improved. And also, a lot of these methods have kind of analogs in ecological network inference. So finally, my conclusions are that at the start of my postdoc, I took a little bit of a risk deciding to be 100% computational. And I was able to, I really enjoyed collaborating with experimentalists. And I feel like we got a lot further teaming up together. And so that was one of the things that I learned from this research experience. That I hope I've convinced you that ecological and transcriptional regulatory modeling, although they seem very different, can benefit from similar assumptions and mathematical frameworks. And then also that transcriptional regulatory network inference is feasible using ATAC-seq and gene expression data. And although all the methods here I've been talking about immune cells, they would obviously be applicable to any host cell, or if we even had single cell microbial expression. So they're very general techniques. Finally, I'd like to acknowledge a fair number of people. So in the Bonneau lab, I've worked closely with all of these people, especially Zach Kurtz and Christian Mueller, developing Speakeasy, the ecological network inference pipeline. And then in the Littman lab, Jason Hall, and especially Maria Prokofsky, who's pored over the networks that I've generated and basically has been willing to team up with me to generate these data sets and also to do experimental validation. I'd like to thank people at the Simons Foundation. So Nick Carriero has helped me set up my bioinformatics pipelines at the Simons Foundation. Aaron Waters is the one who helped me design the IPython notebook visualization for the network. And also like to thank Aviv Madar, who is the author of the Th17 paper and really helped me working with that data set and doing the benchmarking. All right, and so with that, I'd like to take any questions that you might have.
Yeah, very nice, thanks. For the microbial interaction networks and perhaps the transcriptional networks as well, sequence abundance is typically normalized to library size or subsampled, and so if one tax or gene increases, some other gene or combination has to decrease. So does that dependence interfere with the models, or how do you handle that?
So you do have to handle that, and in Speakeasy, we handle that explicitly. So we borrow a data transformation from mathematical geology. So we use a centered log ratio transformation and basically use compositional data analysis techniques. There are some theoretical results that in a high dimensional, once we do that transformation in a high dimensional setting, when we infer our inverse covariance matrix, it actually is a good proxy for an inverse covariance matrix, which would be based on the absolute abundances, which is information, of course, we don't have. But in a high dimensional setting, using that transformation, we do account for it. So we do expect that our results will be compositionally robust there. With regard to transcriptional regulatory networks and gene expression data sets, compositional biases happen. There's a trade-off between what's known as effective population size, or the entropy of the data set, and whether or not you'll have compositional artifacts. So for example, if you have, so there are several ways that you could increase the entropy of, we'll call a microbial abundance data set. So you increase, so entropy would basically describe how little you know about something. And so if you had a distribution, so if you had a lot of microbial measurements, that would be you would have a higher entropy. So entropy increases as you have an increased number of states, but it also increases as your probability distribution flattens out. So if you have a high equal probability of being spread, whereas a low entropy distribution, you would have one microbe that was dominating. So it turns out with transcriptional data sets, compositional artifacts aren't a big deal because you have so many genes that you're measuring, and you don't have one dominant gene as you might have, for example, in a microbiome data set. And so we don't account for compositional effects in transcriptional data sets. But in the microbiome world, it's very important.
My question is about the sparsity of the network.
Mm-hmm.
So how sparse it is will also depend on the range of diverse biological perturbation you subject the system to. Sometimes a more severe challenge can make a denser network sparser, or vice versa. I wonder if you've seen architectural changes in your benchmark data set.
Oh, would you repeat the question? Just
so if you didn't challenge the system, just have a very simple steady state data set, you might observe one type of network architecture might be very sparse. But if you push it to a very challenged state, putting together, you might see a lot more connections being made. And therefore, the architecture itself could be predictive of biological states, or the sparsity can be scored as yet another kind of a network characteristics that you can extract.
right.
So I don't know whether your benchmark data set has challenged the system sufficiently diverse to uncover all the potential edges.
So there are definitely limitations with the gold standard knockout chip data, and most likely it is too dense of a gold standard and not representative, because we're looking at the 11 key transcription factors in Th17. And so those transcription factors automatically have a lot of targets. And what we're missing from our gold standard is a transcription factor that doesn't really play a role in Th17 biology. So definitely there's some bias there. That said, we do have a collaboration with Vijay Kutru where we have a TR1 data set. And so he's going to be sharing with us, I think, about 32 knockout experiments. So I'm planning on benchmarking my methods in that system too. But yeah, there's definitely some bias in the benchmark that I used. Mm-hmm.
You have candidate transitional factors in the innate lymphoid cell 3, and then you said you're gonna CRISPR them in the progenitors and put it back to the mouse.
Mm-hmm.
This makes sense, but maybe complicated. The analysis. These cells have to migrate to mucosal tissues that in testing. There will be other cells with similar transitional factors like Th17 cells. There, we don't know how many cells will arrive to these tissues. So this potentially, I see the approach makes a lot of sense, but I'm not sure how realistic this will be to get information, to ask the question, how important are these predicted networks in the biology of the system? You know, this is very challenging, I would think.
These are extremely challenging experiments, agreed. And we've made measurements in adult steady state cells. And so it might be the case that we knock out the transcription factor and the ILC3 doesn't even differentiate into an ILC3. And so, yeah, there's fingers crossed these will work. But I think it's important to have experimental validation of the method. Some of the cell types, so ILC2s can survive a bit in tissue culture. And so this is not our only means, but we were hoping to do some functional validation. We actually have some interesting gene targets in the ILC3s that we know are involved in, for example, ILC3-Th17 interaction. So we have some interesting hypotheses about what kind of physiological effects we'll have.
You were making an assumption about sparsity in your interaction networks, and I guess I can see how that assumption might be valid, but it's
seems like it's especially valid if you include any environmental parameters that may be controlling multiple nodes in your network. So do you ever do that? Do you include environmental parameters as well in these networks
That would be something that should be done. Currently, we've just been working with cross-sectional data, and we haven't been we haven't been accounting for environmental factors, but those would be very important measurements to include.
Sort of related to that question with the microbial networks. So, and I'm never sure how to deal with this. Some people make this hypothesis that there are keystone species in microbial consortia. One would assume that those have a higher degree of connectivity than the others. Would your models be sensitive to that, or do they provide, based on the data sets you have, evidence that perhaps there aren't these highly connected nodes? In other words, the sort of a lack of this idea of a keystone species in the community?
So my collaborators, Christian and Zach, have gone on to apply Speakeasy to pretty much every microbial data set under the sun, looking at soil samples, looking at different body sites. And so they've come up with, they say that the networks are sticky or a small world, and so they do have some hubs. Also, just as a side note, one of the things that really surprised me in the benchmarking of Speakeasy is that the topology of the network actually has huge implications for how easy it is to infer the network. So it's basically the number of samples you need in a theoretical setting. After we saw this empirically, we browsed the high-dimensional statistics literature, and what we saw was that the number of samples that was required to recover a network that was actually generated from a multivariate Gaussian, so a very nice network, was actually directly dependent on the max degree in the network. So this suggests that networks that do have hubs, for example, a scale-free network, are a lot harder to infer than, say, a random network or a network that is cluster-based, where every node is limited to being connected to like 5 other nodes. So max degree does affect inferability.
Thanks so much. So do you think you'll abandon microbial community in